Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In 1953, James Watson and Francis Crick unveiled the double helix structure of DNA, forever changing humanity's understanding of life itself. Their discovery revealed that within every cell lies a four-letter code, adenine, thymine, guanine, and cytosine, that determines everything from eye color to disease susceptibility. Today, nearly 70 years later, scientists stand at the threshold of not just reading this genetic blueprint, but rewriting it entirely. The Human Genome Project, completed in 2003, mapped all 3 billion base pairs of human DNA. This monumental achievement cost nearly $3 billion and took 13 years to complete. Yet today, genetic sequencing can be performed in hours for less than $1,000. This dramatic acceleration has opened doors previously thought impossible, the ability to edit genes with surgical precision. CRISPR-Cas9, discovered in bacteria as an immune system mechanism, has become the molecular scissors of genetic engineering. This revolutionary tool allows scientists to cut DNA at specific locations and insert new genetic material with unprecedented accuracy. Jennifer Doudna and Emmanuelle Charpentier's Nobel Prize-winning work transformed CRISPR from a bacterial curiosity into the most powerful gene-editing technology ever developed. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free, and it helps us grow this community together. The implications are staggering. Scientists have already created genetically modified organisms that produce human, insulin, manufacture spider silk proteins, and even glow in the dark. Plants have been engineered to resist diseases, survive droughts, and produce enhanced nutrients. Animals have been modified to grow human organs for transplantation, yet the ultimate frontier remains largely untouched: human genetic modification. Current genetic therapies focus on treating diseases after birth. Sickle cell anemia, a condition affecting millions worldwide, occurs when a single letter change in the beta-globin gene causes red blood cells to form characteristic crescent shapes. Recent clinical trials using CRISPR have successfully corrected this mutation in patients' bone marrow cells, essentially curing the disease. But what if we could prevent genetic diseases before birth? What if we could enhance human capabilities beyond their natural limits? What if we could design humans resistant to cancer, immune to viral infections, or capable of surviving in extreme environments? These questions thrust us into the realm of designer babies and custom-built humans. The technology already exists in primitive forms. In vitro fertilization allows parents to select embryos free from specific genetic disorders. Pre-implantation genetic diagnosis can screen for over 400 genetic conditions. Some fertility clinics offer sex selection, allowing parents to choose their baby's gender. These practices represent the earliest steps toward genetic customization, yet the ethical landscape remains treacherous. In 2018, Chinese scientist He Jiankui shocked the world by announcing the birth of the first gene-edited babies, twin girls modified to resist HIV infection. The international scientific community condemned his work, leading to his imprisonment and a global moratorium on germline editing, modifications that can be passed to future generations. The technical challenges are equally daunting. Human genetics operates through complex networks, where single genes often influence multiple traits. Modifying one characteristic might inadvertently affect others in unpredictable ways. The concept of genetic enhancement assumes we understand which traits are universally beneficial, a presumption that ignores the diversity that has enabled human survival across millennia. Despite these obstacles, research continues advancing toward human genetic modification. The question is no longer whether we can create custom-built humans, but whether we should, and if so, how we can do it responsibly while preserving human dignity and genetic diversity. Inside every human cell lies a molecular factory more sophisticated than any human-engineered system. This factory operates according to instructions written in DNA, transcribed into RNA, and translated into proteins that perform the countless functions necessary for life. Understanding this process has enabled scientists to develop tools that can rewrite these genetic instructions with remarkable precision. CRISPR-Cas9 functions like a molecular word processor, capable of finding specific genetic sequences and editing them. The system consists of two main components: a guide RNA that locates the target sequence and the Cas9 protein that cuts the DNA. Scientists can program this system to target virtually any genetic sequence, making corrections, deletions, or insertions with accuracy rates exceeding 99%. Recent advances have expanded CRISPR's capabilities far beyond simple cutting. Base editors can change single letters of genetic code without creating double-strand breaks, reducing unwanted mutations. Prime editors can insert, delete, or replace genetic sequences up to 80 base pairs long with even greater precision. These tools have transformed genetic engineering from a crude process of trial and error into refined molecular surgery. Synthetic biology represents the next evolutionary step, moving beyond editing existing genes to creating entirely artificial genetic circuits. Scientists have designed biological oscillators that make cells blink like biological lighthouses, genetic switches that respond to specific chemical signals, and biological computers that process information using DNA instead of silicon. These synthetic circuits can be combined to create increasingly complex biological machines. Gene drives represent perhaps the most powerful and controversial application of genetic engineering. These systems bias inheritance to ensure that modified genes spread through populations faster than natural selection would allow. A gene drive could theoretically spread a genetic modification through an entire species within a few generations, offering unprecedented power to reshape life on Earth. The technical capabilities for human genetic modification continue expanding rapidly. Scientists have developed methods to edit genes in specific cell types at particular developmental stages and in response to external signals. Optogenetics allows researchers to control genetic activity using light, while chemogenetics enables precise genetic control through specially designed molecules. Epigenetic editing offers another avenue for human modification without permanently altering DNA sequences. These techniques modify the chemical tags that determine which genes are active, potentially allowing reversible genetic modifications. Scientists have used epigenetic editors to turn genes on or off in living animals, opening possibilities for temporary genetic enhancements. The delivery of genetic modifications presents ongoing challenges. Current methods include direct injection, electroporation to create temporary pores in cell membranes, and viral vectors that carry genetic modifications into cells. Lipid nanoparticles, used successfully in some COVID-19 vaccines, have emerged as promising delivery vehicles for genetic modifications. In vivo gene editing, modifying genes directly inside living organisms, has achieved remarkable successes. Clinical trials have demonstrated the ability to edit genes in patients' livers, eyes, and blood cells. Some treatments require only a single injection to provide lifelong therapeutic benefits, representing a paradigm shift from traditional medicine's focus on managing symptoms. Multiplexed editing allows scientists to modify multiple genes simultaneously, opening possibilities for complex genetic redesigns. Researchers have successfully edited up to 25 genes in a single experiment, each modification targeted to specific cellular locations. This capability could enable comprehensive genetic makeovers that address multiple traits or diseases simultaneously. The precision of modern genetic tools has reached the level of individual atoms. Cryo-electron microscopy allows scientists to visualize the exact three-dimensional structures of genetic editing complexes, enabling rational design of improved tools. Machine learning algorithms can predict the effects of genetic modifications before they're implemented, reducing trial and error experimentation. Quality control systems ensure the accuracy of genetic modifications. Scientists have developed methods to detect off-target effects, unintended genetic changes, with sensitivity sufficient to identify single incorrect modifications among billions of correctly edited cells. These quality control measures are essential for any application involving human genetic modification. The molecular workshop for genetic modification has matured from primitive tools to sophisticated biotechnology platforms capable of precise, predictable genetic changes. The technical barriers to human genetic modification continue falling, bringing us closer to the reality of custom-built humans. The human genetic code contains approximately 20,000 genes, each representing a potential target for modification. Scientists have identified genetic variants that provide remarkable advantages: resistance to diseases, enhanced physical capabilities, improved cognitive function, and extended lifespans. These natural genetic variations offer a preview of the enhancements possible through deliberate genetic modification. Disease resistance represents the most immediate application for human genetic enhancement. The CCR5 gene, when modified, provides resistance to HIV infection. Approximately 1% of people of Northern European descent carry mutations that make them virtually immune to HIV. Scientists have successfully replicated this resistance in laboratory settings, suggesting the possibility of eliminating HIV susceptibility in future generations. Cancer resistance offers another compelling target for genetic enhancement. Elephants possess 20 copies of the TP53 tumor suppressor gene compared to humans' single copy. This genetic redundancy helps explain why elephants develop cancer at remarkably low rates, despite their large body size and long lifespans. Researchers have explored whether increasing TP53 copies in humans could provide similar cancer protection. Cardiovascular disease, responsible for over 17 million deaths annually worldwide, could potentially be prevented through genetic modifications. Some individuals carry mutations in the PCSK9 gene that result in extremely low cholesterol levels throughout their lives. These people demonstrate dramatically reduced rates of heart disease, suggesting that similar genetic modifications could provide lifelong cardiovascular protection. Physical enhancement through genetic modification extends beyond disease prevention to performance optimization. The ACTN3 gene exists in two variants, one associated with endurance performance and another linked to explosive power. Olympic-level endurance athletes show significantly higher frequencies of the endurance variant, while elite sprinters and power athletes carry more copies of the explosive power variant. Myostatin inhibition represents one of the most dramatic potential physical enhancements. This protein normally limits muscle growth, but individuals with myostatin mutations develop extraordinary muscle mass and strength. Belgian blue cattle, bred for myostatin deficiency, develop muscle mass increases of 20 to 25%. Similar modifications in humans could potentially create individuals with superhuman strength. Cognitive enhancement through genetic modification remains one of the most intriguing and controversial possibilities. The COMT gene affects dopamine metabolism in the prefrontal cortex, influencing working memory and executive function. Individuals with specific COMT variants demonstrate superior performance on cognitive tests, suggesting potential targets for genetic cognitive enhancement. Brain-derived neurotrophic factor, or BDNF, promotes neuronal growth and survival. Enhanced BDNF expression could potentially improve learning, memory, and cognitive flexibility. Animal studies have demonstrated that increased BDNF levels improve performance on various cognitive tasks, supporting the possibility of genetic cognitive enhancement in humans. Sensory enhancement offers fascinating possibilities for human genetic modification. Some individuals possess tetrachromacy, 4 types of color receptors instead of the typical 3, allowing them to distinguish millions more colors than normal human vision permits. Genetic modifications could potentially provide enhanced color vision, night vision capabilities, or sensitivity to ultraviolet or infrared light. The MC1R gene influences pain sensitivity, with some variants providing natural pain resistance. Individuals with specific MC1R mutations require significantly less anesthesia during medical procedures and report lower pain levels throughout their lives. Such genetic modifications could eliminate chronic pain conditions or provide enhanced pain tolerance. Longevity enhancement represents perhaps the ultimate genetic modification goal. Centenarians and supercentenarians often carry genetic variants that protect against age-related diseases and promote cellular maintenance. The FOXO3 gene associated with exceptional longevity influences cellular stress resistance and DNA repair mechanisms. Telomerase activation offers another pathway to genetic longevity enhancement. Telomeres, protective DNA sequences at chromosome ends, shorten with age and cellular division. Enhanced telomerase activity could potentially maintain telomere length indefinitely, dramatically extending cellular and organismal lifespans. Metabolic optimization could provide enhanced energy efficiency and endurance. Some individuals carry genetic variants that improve mitochondrial function, providing superior energy metabolism and reduced fatigue. Genetic modifications targeting metabolic pathways could potentially create humans with enhanced physical and mental endurance. The catalogue of potential human genetic enhancements continues expanding as scientists discover new beneficial genetic variants and develop improved modification techniques. Each enhancement represents both tremendous opportunity and significant responsibility in shaping humanity's genetic future. The human genome represents 4 billion years of evolutionary refinement, a complex system where individual genes rarely operate in isolation. This intricate genetic architecture creates significant boundaries that limit the possibilities and safety of human genetic modification. Understanding these biological constraints is essential for realistic assessment of genetic enhancement potential. Pleiotropy, the phenomenon where single genes influence multiple traits, represents one of the most significant challenges in genetic modification. The sickle cell anemia mutation, while causing a devastating blood disorder, also provides protection against malaria. This genetic trade-off demonstrates how seemingly beneficial modifications can carry hidden costs that emerge only under specific circumstances. The APOE gene illustrates pleiotropy's complexity particularly well. The APOE4 variant increases Alzheimer's disease risk, but may provide advantages for cognitive performance in young adults and enhanced pathogen resistance. Eliminating APOE4 to prevent Alzheimer's disease could inadvertently reduce other beneficial traits, highlighting the difficulty of genetic optimization. Polygenic traits, characteristics influenced by hundreds or thousands of genes, present enormous challenges for genetic modification. Human height involves over 700 genetic variants, each contributing small effects. Intelligence appears even more complex, with thousands of genetic variants identified, each accounting for less than 1% of cognitive variation. Modifying polygenic traits would require simultaneous editing of numerous genes, exponentially increasing complexity and risk. Genetic networks operate through intricate regulatory systems that control when, where, and how much genes are expressed. The same gene may be active in multiple tissues at different developmental stages, performing distinct functions in each context. Modifying such genes could improve one function while disrupting others, creating unintended consequences that might not appear until years or decades later. Epigenetic regulation adds another layer of complexity to genetic modification. Chemical modifications to DNA and associated proteins control gene activity without changing the underlying genetic sequence. These epigenetic marks can be influenced by environmental factors, age, and life experiences, making the effects of genetic modifications potentially variable and unpredictable. Gene dosage sensitivity presents critical constraints for genetic enhancement. Many genes require precise expression levels to function properly. Too little causes deficiency, while too much can be equally harmful. The MECP2 gene exemplifies this principle. Insufficient expression causes Rett syndrome, while overexpression leads to autism-like symptoms. Genetic modifications must achieve precise dosage balance to avoid unintended consequences. Evolutionary trade-offs reveal that many genetic variants represent compromises rather than pure advantages. The genetic variants associated with larger brain size in humans also increase the risk of psychiatric disorders. Enhanced immune responses that protect against infections can increase autoimmune disease susceptibility. These evolutionary balances suggest that genetic improvements may inevitably involve trade-offs. Developmental timing creates additional constraints on genetic modification. Many genes play critical roles during embryonic development, but have different functions in adult tissues. Genetic modifications might need to be precisely timed to achieve desired effects without disrupting normal development. This temporal complexity makes genetic enhancement significantly more challenging than simple gene replacement therapies. Genetic background effects mean that the same genetic modification can produce different results in different individuals. A genetic variant that provides benefits in one ethnic population might be neutral or harmful in another due to interactions with other genetic variants. This genetic context dependence complicates the development of universal genetic enhancements. Population. Genetics principles reveal additional boundaries to genetic modification. Genetic diversity provides resilience against environmental changes and disease outbreaks. Excessive genetic homogenization through widespread adoption of similar enhancements could reduce human genetic diversity, potentially making our species more vulnerable to future challenges. Mosaicism, the presence of genetically different cells within the same individual, can result from genetic modifications that don't reach every cell. This cellular heterogeneity can lead to unpredictable effects and reduced efficiency of genetic enhancements. Achieving complete and uniform genetic modifications throughout the human body remains technically challenging. Long-term stability of genetic modifications presents ongoing concerns. Some genetic changes might be reversed by cellular repair mechanisms or lost during cell division. Others might accumulate additional mutations over time that alter their effects. Ensuring permanent and stable genetic modifications requires understanding and overcoming these cellular processes. The biological boundaries of genetic modification remind us that the human genome represents a delicately balanced system shaped by millions of years of evolution. While genetic enhancement holds tremendous promise, these natural constraints require careful consideration to avoid unintended consequences that could compromise human health and genetic diversity. The power to reshape human genetics forces humanity to confront profound ethical questions that strike at the core of what it means to be human. These moral considerations extend far beyond individual choice, encompassing societal justice, human dignity, and the rights of future generations who cannot consent to the genetic modifications they inherit. Germline editing, modifications that can be passed to offspring, presents the most ethically complex scenario. When scientists alter the genetic code of embryos, sperm or eggs, these changes become permanent parts of the human gene pool, affecting not just the modified individual, but all their descendants. This irreversible modification of human heredity raises questions about whether any generation has the right to make genetic decisions for all future generations. The concept of informed consent becomes particularly challenging in genetic modification contexts. How can parents provide meaningful consent for genetic modifications whose long-term effects remain unknown? How can future generations consent to genetic changes made before their birth? Traditional medical ethics, built around individual autonomy and informed consent, struggles to address modifications that affect people who don't yet exist. Equity and access represent critical ethical concerns in human genetic modification. If genetic enhancements become available but remain expensive, they could create or exacerbate social inequalities. Enhanced humans might possess significant advantages in education, employment, and life opportunities, potentially creating a genetic class system where genetic modifications determine social status. The enhancement versus treatment distinction has sparked extensive ethical debate. Most ethicists accept genetic modifications to treat serious diseases, but genetic enhancements that improve normal human capabilities raise different moral questions. Where does treatment end and enhancement begin? Is preventing genetic predisposition to heart disease treatment while improving memory capacity enhancement? These definitional challenges have profound implications for policy and regulation. Cultural diversity adds complexity to genetic modification ethics. Different societies hold varying values regarding disability, human perfection, and the desirability of genetic modifications. What one culture considers a beneficial enhancement, another might view as unnecessary or even harmful. Global governance of genetic modification must navigate these cultural differences while protecting fundamental human rights. The commodification of human genetics presents disturbing possibilities. If genetic modifications become commercial products, will human genetic diversity be reduced to market preferences? Could genetic enhancement lead to the commercialization of human reproduction, where parents select genetic traits like consumer products? These concerns reflect deeper questions about the relationship between market forces and human dignity. Genetic discrimination represents a significant ethical and practical concern. Even if genetic modifications provide benefits, they might also create new forms of discrimination. Employers, insurers, or educational institutions might favor genetically enhanced individuals, creating systematic disadvantages for those without modifications. Legal protections against genetic discrimination would need to evolve alongside genetic modification technologies. The perfectibility versus diversity tension highlights fundamental philosophical differences about human nature. Some argue that genetic modifications represent humanity's natural drive toward improvement and perfection. Others contend that human diversity, including genetic variations that cause disabilities, represents inherent human value that should be preserved rather than eliminated through genetic modification. Religious and spiritual perspectives on genetic modification vary widely. Some faith traditions view genetic enhancement as fulfilling divine mandates to heal the sick and improve human welfare. Others see genetic modification as inappropriate interference with divine creation or natural order. These theological differences influence public opinion and policy regarding genetic modification research and applications. Children's rights present unique ethical challenges in genetic modification contexts. Do children have the right to genetic modifications that could improve their lives? Do they have the right to an unmodified genetic inheritance? How do we balance parental decision-making authority with children's future autonomy? These questions become particularly acute when considering irreversible genetic modifications. Justice considerations extend beyond individual fairness to global equity. If genetic modifications become available primarily in wealthy nations, could they exacerbate global inequalities? Should genetic enhancement technologies be considered global public goods that require equitable international distribution? These questions connect genetic modification ethics to broader issues of global justice and development. The slippery slope argument suggests that accepting certain genetic modifications could lead inevitably to more extreme enhancements. Critics worry that therapeutic genetic modifications could normalize genetic enhancement, eventually leading to eugenic practices or the creation of genetic castes. Supporters counter that careful regulation and ethical oversight can prevent such extremes while allowing beneficial applications. Professional responsibility creates ethical obligations for scientists, physicians, and policymakers involved in genetic modification research and applications. These professionals must balance scientific advancement with public safety, individual welfare with societal good, and present benefits with future risks. Professional codes of ethics continue evolving to address these unprecedented responsibilities. The ethical labyrinth of human genetic modification reflects humanity's struggle to responsibly manage transformative technologies. These moral considerations will ultimately determine whether genetic modification becomes a tool for human flourishing or a source of new forms of inequality and injustice. The international response to human genetic modification has been fragmented and evolving, reflecting the tension between scientific advancement and ethical concerns. Different nations have adopted varying regulatory approaches, creating a complex global landscape where genetic modification research and applications face dramatically different legal and cultural constraints. The United Kingdom established one of the most comprehensive regulatory frameworks through the Human Fertilization and Embryology Authority, created in 1990. This organization oversees embryo research, including genetic modifications, through a licensing system that balances scientific progress with ethical oversight. The UK permits embryo research up to 14 days of development and has approved limited clinical applications of mitochondrial replacement therapy. Germany maintains some of the world's strictest restrictions on genetic modification research, largely influenced by historical concerns about eugenics. The Embryo Protection Act prohibits germline modifications and restricts embryo research, reflecting deep cultural sensitivity to genetic manipulation. These restrictions have limited German contributions to genetic modification research while raising questions about scientific competitiveness. China's regulatory environment has been marked by rapid changes following the He controversy in 2018. Initial permissive attitudes toward genetic research gave way to stricter oversight after international condemnation of the first gene-edited babies. New regulations require approval from national health authorities for clinical trials involving genetic modifications, representing a significant tightening of oversight. The United States presents a complex regulatory landscape involving multiple agencies. The Food and Drug Administration oversees clinical trials of genetic therapies, while the National Institutes of Health sets research funding policies. Congressional appropriations bills have historically prohibited federal funding for germline editing research, though private research remains less restricted. This mixed approach has allowed continued research while limiting certain applications. Japan has emerged as a leader in regenerative medicine and genetic modification research through supportive regulatory frameworks and significant government investment. The country has approved clinical trials for various genetic therapies and maintains relatively permissive policies for basic research while implementing careful oversight for human applications. International organizations have struggled to develop unified approaches to genetic modification governance. The World Health Organization established an expert advisory committee on developing global standards for governance and oversight of human genome editing in response to the He incident. However, this committee's recommendations remain non-binding, highlighting the challenges of international coordination. Scientific diplomacy has become increasingly important as nations recognize the global implications of genetic modification research. International conferences, professional organizations, and collaborative research agreements provide forums for sharing information and developing common standards. However, these voluntary mechanisms have limited enforcement power when nations choose different regulatory paths. The concept of genetic sovereignty has emerged as nations consider their regulatory approaches. Some countries view genetic modification technologies as matters of national security and economic competitiveness, leading to policies that prioritize domestic research and development. This nationalism could fragment global research efforts and create regulatory competition rather than cooperation. Brain drain and research migration have become significant concerns as scientists move between countries with different regulatory environments. Restrictive regulations in some nations have driven researchers to more permissive jurisdictions, potentially concentrating genetic modification expertise in countries with fewer ethical constraints. This migration could undermine international cooperation and equitable development of genetic technologies. Patent and intellectual property considerations add complexity to international genetic modification governance. Different patent systems and enforcement mechanisms create varying incentives for research and development. Some argue that genetic modifications should be treated as global public goods rather than proprietary technologies, while others contend that patent protection is essential for innovation. Trade implications of genetic modification technologies are becoming increasingly apparent. Nations with advanced genetic modification capabilities might gain significant economic advantages in healthcare, agriculture, and biotechnology sectors. Trade agreements increasingly include provisions related to biotechnology and genetic modification, reflecting their growing economic importance. Regulatory arbitrage, the practice of seeking jurisdictions with favorable regulations, poses challenges for global genetic modification governance. If some countries maintain permissive regulations while others impose strict restrictions, research and applications might concentrate in less regulated areas. This fragmentation could undermine global standards and create ethical concerns about exploitation. International clinical trials present particular governance challenges when genetic modifications are involved. Different national regulations can complicate multi-country studies, while varying ethical standards raise questions about participant protection. Harmonizing international clinical trial regulations for genetic modifications remains an ongoing challenge. The emergence of genetic tourism, traveling to other countries for genetic treatments not available domestically, parallels earlier trends in reproductive medicine. Patients might seek genetic modifications in countries with more permissive regulations, potentially undermining domestic policy goals and creating safety concerns for inadequately regulated treatments. Capacity building in developing nations represents both an opportunity and a challenge for global genetic modification governance. While technology transfer could provide access to beneficial genetic therapies, inadequate regulatory infrastructure might create safety risks. International efforts to strengthen regulatory capacity in developing nations are essential for equitable and safe global development of genetic modification technologies. The global response to human genetic modification continues evolving as technologies advance and experience accumulates. Achieving effective international coordination while respecting national sovereignty and cultural differences remains one of the greatest challenges in governing these transformative technologies. The convergence of genetic modification, artificial intelligence, and biotechnology is reshaping the trajectory of human evolution itself. For the first time in our species' history, we possess the tools to direct our own genetic development, potentially creating forms of humanity that would be unrecognizable to previous generations. The decisions we make in the coming decades will determine whether these technologies become instruments of human flourishing or sources of unprecedented division. Artificial intelligence is accelerating genetic modification research at an unprecedented pace. Machine learning algorithms can analyze vast genetic datasets to identify beneficial mutations, predict the effects of genetic modifications, and design optimal genetic interventions. DeepMind's AlphaFold system, which predicts protein structures from genetic sequences, has revolutionized our understanding of how genetic modifications might affect cellular function. The timeline for widespread human genetic modification appears to be accelerating rapidly. Current clinical trials for genetic therapies are already treating thousands of patients with modified genes. Within the next decade, genetic modifications for serious diseases will likely become routine medical procedures. Genetic enhancements for healthy individuals might follow within 20 to 30 years, depending on regulatory approval and public acceptance. Convergent technologies are creating possibilities that extend far beyond simple genetic modifications. Brain-computer interfaces combined with genetic modifications could create enhanced cognitive capabilities that merge biological and artificial intelligence. Synthetic biology might enable the creation of entirely artificial genetic circuits that provide humans with capabilities not found in nature. The democratization of genetic modification technologies could make these tools accessible to individuals and small organizations rather than just major institutions. CRISPR kits are already available for purchase online, and biotechnology costs continue declining rapidly. This accessibility raises both opportunities for innovation and concerns about safety and regulation. Space exploration presents unique opportunities and challenges for human genetic modification. Long-duration space travel exposes humans to radiation levels that could cause genetic damage over time. Genetic modifications that enhance radiation resistance, bone density maintenance, and adaptation to low gravity could become essential for interplanetary exploration and colonization. Enhancement cascades represent a potential future where genetic modifications become increasingly common and sophisticated. As some individuals receive genetic enhancements, competitive pressures might drive others to seek similar or superior modifications. This dynamic could lead to rapid genetic divergence within human populations, potentially creating distinct genetic classes or even subspecies. The integration of genetic modifications with other enhancement technologies could create hybrid human-machine beings. Genetic modifications that improve compatibility with neural implants, enhanced healing to support cybernetic integration, or modified metabolism to power artificial organs could blur the boundaries between biological and technological enhancement. Reversibility and upgradability of genetic modifications present both opportunities and challenges for the future. Developing methods to reverse or modify existing genetic changes could make genetic enhancement more acceptable by reducing permanent consequences. However, the ability to continuously upgrade human genetics might create pressure for constant modification and genetic obsolescence. Global genetic governance will likely require new international institutions and agreements specifically designed for genetic modification oversight. These organizations would need to balance scientific advancement with safety, equity, and ethical concerns while respecting national sovereignty and cultural differences. The development of effective global governance mechanisms will be critical for managing genetic modification technologies responsibly. Economic disruption from genetic modification technologies could rival or exceed the impact of artificial intelligence on employment and social structures. Enhanced humans might have significant advantages in many fields, potentially displacing unmodified workers. Social support systems and economic structures might need fundamental reorganization to accommodate genetic enhancement's effects on human capabilities and opportunities. The preservation of human genetic diversity will become increasingly important as genetic modification technologies advance. Maintaining genetic variation within human populations provides resilience against environmental changes and disease outbreaks. Policies and practices that encourage genetic diversity while allowing beneficial modifications will be essential for long-term human survival. Ethical evolution will need to keep pace with technological advancement. Our moral frameworks, developed for unmodified humans in pre-technological societies, may prove inadequate for addressing the challenges of genetic enhancement. New ethical systems that can guide genetic modification decisions while preserving human dignity and rights will be essential. The ultimate question facing humanity is not whether we can create custom-built humans, but whether we should, and if so, how we can do it responsibly. The technologies exist today to begin modifying human genetics in profound ways. The choices we make about their development and application will determine whether genetic modification becomes a tool for human flourishing or a source of new forms of inequality and conflict. James Watson and Francis Crick's discovery of the double helix structure launched a scientific revolution that has brought us to this pivotal moment in human history. We now possess the ability to rewrite the genetic code that has guided human development for hundreds of thousands of years. The future we're building through genetic modification will reflect our highest aspirations and deepest values about what it means to be human. The transformation has already begun. Every day, researchers make new discoveries that expand the possibilities for human genetic modification. The question is no longer whether we will create custom-built humans, but what kind of humans we will choose to become. That choice and its consequences will define the future of our species.